Hello friends, the next topic we will see is the plasma expanders. The learning outcome for this chapter is that on completion of these sessions, the learner will be able to define and describe the blood plasma expander, what these are the blood plasma expander. Then they can describe the role and need of a plasma expander. They can describe the various type of plasma expanders and what are the ideal properties of a plasma expander and application of the plasma expanders in the pharmacotherapy. So before going to the plasma expander, first of all we will see what is a blood, then blood plasma, and then we will see the plasma expanders. So as far as the blood and blood plasma, we have already seen in the course of APHE. Blood is a fluid connective tissue which is circulated continuously around the body, and it is one of the communication. It does the communication between the tissues which are placed at the distant place, like an endocrine system. From endocrine system, it transport the hormones to the various tissue, and from the various tissue, it transport the waste product to their excretory organs. So it act as a transportation, communication, and all these things we have already seen. Now, what is a plasma? Plasma is nothing but a straw color watery liquid in which several blood cells are suspended. We have already seen these terms in the EPG. So, blood minus your blood cells, remaining fluid is a plasma. And now the term plasma expander, it is actually the important. It is a relatively high molecular weight substances which boost the plasma volume and increases the osmotic pressure. So here expanders are the substances which are the substitutes of the plasma. They are not the substitute of the blood. So remember well. So blood plasma and expander. So expander substitute the plasma. They are the high molecular weight substances which boost the plasma volume and they restores the osmotic pressure. So they are used in the treatment of the patients which are suffering the hemorrhagic or any kind of a shock where the plasma volume is dropped down, or to restore the plasma. So IV fluid also they are increased. So retention of a fluid volume and increasing in the fluid. So these are the replacement of a lost fluid due to the illnesses, trauma, surgery. These are the specific purpose of giving the plasma expander. They also correct the hypovolemia due to the loss of plasma or blood. So due to the loss of plasma, there is a decrease in the volume, and to counteract or to correct this hypovolemia, the expanders will be given. What are the ideal properties? What should be the ideal properties of this plasma expander? So the very first property, they should be isotonic with the blood. This is the most ideal property of the plasma expander. It should be distributed to the IV compartment only. It should not go anywhere. It should not be distributed inside the body. So it should retain into the IV compartment, because it is for the plasma. It is a replacement of the plasma. It is not a replacement of all other fluids which are present in our body. So it should be distributed only in the IV compartment. Pharmacodynamically, it should be inert. It should not have any pharmacological effect as such. It should be non-pyrogenic, non-allergic, and non-antigenic. It should not have these plasma expander substances should not have this property, anti-pyrogenic, increase in elevation in the body temperature. They should not produce any kind of allergic reaction or antigen antibody type of reaction. They should also be such that they should not be specific to the particular blood group, so non-interfering with the blood flow and cross-matching. Like whenever anything which we are giving to the blood, we are matching with the blood group and blood typing, cross-matching we are doing. So they should not interfere any kind of this type of terms. Okay, they should be stable, easily sterilizable, and cheap, because we are inserting them inside the blood, so they should be sterilized properly. So they should have the property to easily sterilizability, and they should be stable whenever they will be sterilized, and they should have a sufficient low cost. Now there are two type of plasma expanders, crystalloids and colloids. Crystalloids are the aqueous solutions of mineral salt. Crystalloids are the aqueous solutions of mineral salts and other water soluble substances like normal saline, sodium chloride saline, or dextrose solution or Ringer solution. So they contains the mineral salts which are water soluble in that. So they increases the plasma volume and produces less peripheral edema. That will be producing the less peripheral edema production is very less. So they required in a very small value, and when given by the intravenous route of administration, they have a very half-life of 3 to 6 hours only. So this is what the typically properties of crystalloids. So crystal solutions we can say. Okay. Next is the colloids. Colloids, these are the large insoluble molecules such as dextran, human albumins or gelatin or as such blood as such. So they are inexpensive. They are a little bit expensive in use as compared to crystalloids, and they maintain the fluid volume. The maintenance of a fluid volume is more effectively as that of crystalloids by the colloids. So one of the typical advantage of a colloids is that they restore the third space loss whenever there is a loss of blood. So mostly that intravenous they retain inside the intravenous and they do not distribute anywhere, and therefore they restore the space very easily and effectively. So intravenous half-life of these agents are 20 to 30 minutes. Half-life is little bit less. What are the agents which are used as a plasma expanders? Normal saline, lactate ringer, dextrose, these are the crystalloids. Then human albumin, dextran, these are the colloids. So work on these both, whether they are crystalloids or colloids, they work on a principle of osmosis. So they draw the water and they increases the plasma osmotic pressure and draw the water inside they draw the water inside the plasma. So they take up the water, and the lost blood volume is restored. The flow is more easier. The blood flow is from the blood vessel is more easier. So they restored the plasma by drawing the water, and the lost plasma volume will be restored, and thereby the viscosity of the blood is maintained and the blood flow as a blood inside that blood vessel will be more easier. So when used when blood and blood plasma are lost, they move to the extravascular compartment like burn, hypovolemia, trauma. So they will be restored. So during the burning or hypovolemia, what happens? The blood and blood plasma moves to the extravascular compartment. So they will be drawn by the osmotic pressure by using these. So they are the temporary measures. Always remember that these plasma expanders are temporary measures to restore the plasma. They do not have any kind of a blood cells. So the functions which are done by the blood cells will never be replaced by. So they do not have any oxygen carrying capacity or coagulation property or like that way. So they are the replacement of the plasma volume only, and they are therefore a temporary measure. The main contraindications behind the plasma expenditures that they may produce the allergic type of reactions, heart failure, anemia, thrombocytopenia. Pulmonary edema is one of the major contraindications with the plasma expander, and they may have the renal insufficiency as one of the major problem. So these are about plasma expanders. If you have any kind of a difficulty, you can contact me at the given number or email ID. Thank you very much.